Here are my disclosures. None of this stuff that I'm going to talk about is relevant to this particular work with these various different entities. So, questions that I want to address. So, do older people living with HIV have unique unmet clinical needs that could be or should be or might be addressed with a cure? And related to that, is the reservoir, the amount of virus that exists in people during long-term ART, is there any evidence that that's harmful in young or old? Does age affect the reservoir directly, and if so, how? And could that affect cure therapies that are emerging? A big concern, not a lot of data, won't spend much time on it, but will the impact of age on immune function, which has been discussed about here, there's a lot of less immune dysfunction, there's global proliferation, there's more inflammation that we see in elderly people. Will this have an impact on these therapeutic strategies? And finally, will we see synergies? Can we see synergies when you look at work being done at NCI, NIA, NHLBI, NIID, all the institutes, all the players, to see if we can actually come up with a unified way of approaching some of these diseases, because there's actually a fair amount of similarities. All right, so the question I always get as a cure person is, why are we doing this, right? Why do we actually need a cure? If you take one pill once a day, generally pretty safe, you'll live forever. And a question that you guys might want to know, are there particular needs among those people living with HIV? So this figure, or another version of it's been shown multiple times here, the sort of pillars of aging, the hallmarks of aging. You know, it's really remarkable when you look at that original review. This is not the original review paper, the one that was published in Cell years ago. When you look at the factors that are known to be associated biologically with aging, there are a lot of stuff that emerges that are things that we as clinicians, people or scientists working in HIV are aware of. But there are some that are very specific that I think that are associated with aging that might specifically affect people who are older. One is stigma. I think that's broad across, but it's interesting. If you do a survey of people with HIV, and the surveys that were done were primarily in older gay white men who've been infected since the 80s and 90s, and you ask them, what is the thing that's affecting your life the most that a cure could deal with? It's stigma. It's the concept that you're HIV positive and you're taking pills every day. So and that's what they want from a cure. They don't want to be HIV positive, and they certainly don't want to be taking pills every day because it's associated with stigma, which, as everyone in this room, can have a huge effect on health, particularly as aging. Polypharmacy is a given. Most patients are on 3, if not more, drugs as a consequence of their HIV, and our toxicity. And toxicity, you know, the dogma in the field is that, yes, indeed, the old drugs were toxic, but the new drugs are benign. And I was sort of in that ballpark. But every time you turn around, you're hearing, even with the new stuff, the integrase inhibitors in particular, but also tenofovir and now TAF, that there are these associations between being exposed to these modern drugs for a prolonged period of time and unexpected side effects, right? And this is a beautiful study done in South Africa that showed that young women, not old people, but young women, but were particularly susceptible on the current highly recommended regimen of FTC, TAF, and dolutegravir to gain weight. And in fact, young women in this study over 2 years gained 10 kilograms, and the investigators believe that that's going to continue to rise. And as I think Carrie mentioned yesterday in her overview from NA Accord, clearly in North America, the same signals are emerging, and in North America, we're seeing these associations between weight gain and incident diabetes. So, long-term ART is not benign. It's certainly better than no treatment. The drugs we have now are better than they used to be, but life without medications would be a good thing. The other big reason I think that we want to particularly think about the unique needs of people who are older has to do with the immune system. If you characterize the immune system in people in their 60s, 70s, 80s, 90s, clearly there's this concept of immunosenescence. T cells don't function well. You get a CD4-CD8 ratio changes. You've got this chronic inflammation. You've got this clonal expansion of these effector cells. The thymus doesn't work. There's no naive cells. There's no new antigens being presented. All that stuff appears probably later than we assume, 70s and 80s, and certainly is going to have an impact. So what do we know about that in the context of treated HIV infection? Well, first of all, it's important to know that when you go on antiretroviral drugs and you're on it for decades, the virus in the plasma goes down typically to about 1 copy or less per ml. The virus is not replicating at a level that we think causes resistance, but it's not gone. If you put a needle in a lymph node, or if you put a needle in a gut tissue, you can often find the virus, and you can often find little islands of virus activity in which the virus RNA is being produced. There's localized activation. There may be cell-to-cell spread, but there is, during therapy in these tissues, these small niduses of inflammatory burst of HIV production. It's not systemic, but it's happening, and it's happening in those tissues where the immune system is supposed to learn how to deal with things and train itself. And so there are reasons to believe that this active reservoir in the context of treatment could be inflammatory and have consequences. And in fact, when we did this study, and it's kind of a complicated slide, I'm not going to get too much into the details here, but on the left, what we did is we took people who were on long-term treatment, suppressed for a while, and we looked in the rectal tissue, and we looked for protein production from the virus, P24, okay? And we don't find it in HIV negatives. We find it in people who are on antiretroviral therapy and doing well. We find a lot of it in people who are on antiretroviral therapy and not doing well in terms of the CD4 counts, those CD4 non-responders, a very, very common phenotype in the elder population, and so forth. And we see these association between the amount of P24 in the blood, in the cells, in the tissues, and the inflammatory response. So collectively, these data would suggest that the reservoir during long-term ART is active, it's producing inflammatory proteins, and it may, as a consequence, have effects on health. But are there any data along those lines? Well, there are two pieces of information. One is an anecdote, which I think is really, really provocative. But we were in this collaboration with Tim Schacker's group at the University of Minnesota, where we sent him rectal biopsies from about 100 people in our cohort in San Francisco. Untreated people, treated people, elite controllers, and the Berlin patient. The Berlin patient obviously being a high interest because he was cured. Tim Schacker did all the stuff blinded and called me up and said, hey, this guy that you said is HIV positive is HIV negative. And I said, how do you know that? He says, because everybody who has HIV has tissue fibrosis at some degree. But this guy, this PID XYZ, has none. I think he's HIV negative. And I said, well, that's interesting. Because he is. And so you think about it, what does that mean? That means that Timothy Brown, the Berlin patient, had been on ART, had virus production, fibrosis. He was cured. The virus was removed from the equation. Many, many years later, no fibrosis. The implication is that the virus being produced during treatment has an impact on tissue fibrosis, which is a major player in immune dysfunction and chronic inflammation. So there's that anecdote, which I adore, because I like my anecdotes. And then there's these cohort studies, which other people probably respect more, and these are a little bit sloppy, but and some were done by people in this room, including I think Scott must have been involved in some of this stuff. But there have been these studies that have been looked at whether or not, in the context of long-term therapy, the amount of HIV DNA in circulating bloods is a predictor of morbidity and mortality. And there have been a couple studies suggesting that there might be a relationship between the amount of DNA and neurocognitive decline. And work that we have not published, that we've done with Priscilla Hsu, suggests that the amount of virus, the HIV DNA levels, the reservoir, the more that you have, the more likely you are to progress to some cardiovascular endpoints. So these comorbidities that we often associate with aging may be related to the size of the reservoir, suggesting that, that the HIV that persists during therapy is causing immune dysfunction, tissue fibrosis, and bad things. And hence, if we want to restore health to those people who are older, we need to remove treatment and the virus. So I think there's a unique need for a cure in this setting. But there are problems, right? In HIV-infected individuals, as they get older, don't do as well in terms of their capacity to reconstitute a normal immune system. And this is not surprising. If you look in the HIV uninfected population, there is this concept that with age, CD4 counts decline, CD counts increase, and the immune system doesn't work as well. And the reason why I bring this out is that most of the work that we're doing in the cure arena ultimately involves going into a person and interrupting or enhancing a biologic pathway associated with immune function. And if your immune system does not work, you are therefore less likely to respond or more likely to get a bad reaction. So all the clinical trials that we're doing generally require people to be less than the age of 65. And I am, Jeff Taylor, are you still here? So Jeff and I have been involved in these discussions. And Jeff, are you a fan of the 65 or not? You what? Hell no, good. Okay, we got it. Because I have been fighting the fight against the powers that be to release us from this artificial number of 65. I do think age is important, but it's not chronologic, it's biologic. And perhaps we can't define biologic age, but just to routinely exclude people from over the age of 65 takes away the people who have the most altruism and the most desire, the most motivation, in my experience, to be in these studies. And also, I don't think it's scientifically valid. So anyway, that's an issue that's preventing us from studying these things in older individuals. All right. So what is the any impact that age has on the immune system, and might that affect the reservoir? All right. So this is a beautiful study done by Donna Faber and her team, I think at Cornell here in New York. Anyway, they did these various different autopsy studies in people over periods of decades, and they quantify and they characterize in blood and tissues what the T cells look like, CD4 and CD8s. And as you can see here, as you get older, your naive T cells drop, your thymic output drops, and eventually you end up with this expanded population of largely effector CD4 cells that perhaps don't function as well, and so forth. And Alan Landay and his team has actually shown that in the context of HIV infection, this is happening, but it might be accelerated by 1 or 2 or more decades. So this concept of immunosenescence, that you get these effector cells, which are less functional, is one that is borne out in all sorts of studies and is basically now accepted as fact. These cells in the older people tend to basically, particularly in the effector cell population, express markers of activation, expect markers of homeostatic proliferation. Now, why is this important? And another related fact is that if you take an older person as they get into their 60s and 70s, and you observe what's happening with their immune system as that's being reshaped so that you have this massive expansion of effector cells, and if you throw CMV into that situation, and half of the world's population is CMV positive, but 99% of the men in my clinic are CMV positive. If you throw CMV in there, you get this nuclear bomb effect, and you get this massive expansion of your CD8 and also CD4 cells, so that by the time you're in your 60s and 70s, 80s, big chunk of your immune system are these clonally proliferating effector cells. Okay? Why is that important? Well, that's important because every time you turn around in the literature, there's a high-impact paper showing that essentially the reason why HIV persists in people is that it gets into these effector cells, it's at rest in these effector cells, it's not expressed in these effector cells, and as those effector cells clonally expand and populate and proliferate, so does the reservoir. So although it has not been studied, and I think we're going to have a grant soon to look at this, one would propose that as people get older, the reservoir size will be in this population increasingly and perhaps getting bigger and bigger and bigger. So I am pretty sure that age will have an impact on where the virus is, how active it is, and how long it's going to persist. And sort of along these lines, we were beginning carefully to look at factors associated with it, whether you have a large reservoir or a low reservoir, and one that constantly pops up is the CD4-CD8 ratio, which is actually one of these markers of sort of immunosenescence. And basically, if you have evidence biologically of having one of these immune systems that's not working as well, you have a higher reservoir. So it all sort of makes sense, but to be honest, it's poorly studied. And if anyone has any grants that want to get involved in this work, we have the cohort, we just don't have the funds sometimes to answer some of these questions. So, inflammation, right? There's been a lot of discussion about chronic inflammation. HIV-infected people have chronic inflammation. It goes down during ART. It does not normalize. It predicts bad things. Same thing in older individuals. Older individuals have a more of a chronic inflammatory state, particularly if they're CMV seropositive. It's not good for you, predicts bad things. How does this affect the HIV agenda? Well, you know, it's unknown, but here's just one of many papers, but this is our team is actually becoming very, very engaged in imaging work, this because the reservoir is largely in tissues. It's hard for us to get at it, so we're really doing a lot of work with imaging. And when you look at markers of inflammation during long-term treatment, certain areas light up, particularly the lymph nodes. Lymphoid tissues have this persistent evidence of immune activation, inflammation, years into ART. And that's where the virus is, and that's where the immune system is doing what it needs to do. And if you look more carefully at which cell types harbor the virus, you often see these modest, they're not dramatic, but these modest enrichment of HIV in cells that express markers of activation, PD-1, TIGIT, HLA-DR, and so forth. So you have this chronic inflammatory environment. You have these cells that are inflamed, they're proliferating. The virus is modestly enriched in this. So it's probably a problem. I think it's a problem because of this, though. I mean, this is a concept that is essentially at the core of the cancer immunotherapy revolution. That is that you have in cancer tissues, as you have in HIV tissues, you have a chronic inflammatory environment related to the pathogen or the cancer. As a consequence of this, you get upregulation of all these immunosuppressive pathways. You know, inflammation occurs, but what happens then is this overreaction for the immune system to shut everything down. So you end up with an immunosuppressive environment where T cells don't work well, and a lot of the cancer therapies and increasingly a lot of the cure therapies are aimed at blocking those pathways. So I think chronic inflammation will need to be addressed specifically, and I think it's going to be probably a bigger problem in older people because it's more of an inflammatory environment in order to get the T cells and everything else we want to work. So, how might we go about doing that? So let me just tell two stories, and then I'll be done. And the point of these stories is to describe how I think if the powers that be, and mainly it's NIH, can somehow enable the NIA and the geroscientists, the oncologists for sure, probably NHLBI working on inflammation. I mean, there's a just amazing amount of activity going on in these non-HIV diseases that are highly relevant to the cure agenda. And let me just give you one example. So rapamycin, you know, rapamycin is a fascinating drug, Mtor inhibitor. I'm told, and I was at a meeting recently with a bunch of, with some experts in aging, therapeutic approaches to aging. There's this, the people are getting ready for this sort of phase 3 study to see if metformin can enhance lifespan. So it's basically the FDA and people are beginning to think about how to improve health during aging, with health span, not lifespan, sort of being the outcome. In mice, one of the more exciting ways to take healthy mice, not a sick mice, but healthy mice and make them live longer, is to block with low doses Mtor. And this has been repeated and shown in multiple pathways, and it reduces the inflammatory environment. It might have an impact on other pathways, including autophagy. There's a nice poster here about autophagy and aging. And so there's all these different pathways by which blocking Mtor can result in improved health. And in fact, in that syndrome of children having that accelerated aging, these drugs are being studied to see if it can help. The reason why I mention that is that actually, according to my discussions in hallways with the gerontologists who are thinking about the next big study, people are thinking about low-dose Mtor inhibitors as a way to basically improve health in the aged. Dosing is key, because if you give too much, these drugs can be quite toxic. So there's this huge amount of data from the aging literature about how blocking mTOR can enhance health span. It has all these interesting effects on the immune system. It reduces T-cell proliferation, reduces the activation markers, and enhances the capacity of the immune system to basically respond to vaccines, and so forth. And it's quite immunologic active in ways that might be of high interest to people doing cure work. And in fact, and for people who are actually interested in improving immune function. So this was a really wonderful study done. I think it's about 250 older people. And I asked the question, people who are older, they don't respond as well to vaccines. Can we use mTOR inhibitor because of its effect on immune function to enhance vaccine responsiveness? Big clinical trial done with low doses of 2 mTOR inhibitors funded by Novartis. And people were randomized to the mTOR inhibitors or not, and you saw this, significant reduction in infectious complications, mainly the flu, but also in other viral infections and other nonspecific infections. But the other thing that you saw was an increased responsiveness to the vaccine itself. So mTOR's inhibitors as a vaccine adjuvant, as a way to enhance aging, is something that other groups are looking at carefully, because again, it's not necessarily the most benign drug. It all depends on the dose. So we were vaguely aware of this story and some other ones. So we did a cross-sectional study in a transplant population, and we simply asked the question, if you take a group of people who are on ART and they're receiving an mTOR inhibitor to help prevent transplant rejection or not, was the mTOR inhibitor associated with a different reservoir size? And in fact, we saw a modest reduction in the amount of HIV DNA in transplant HIV-infected people post-transplant receiving an mTOR inhibitor. So based on all this, based on everything that's happening in the aging area, in infectious disease, vaccine responsiveness, and our own preliminary data in people, we designed ACTG 53 something. I could never do, I was always a 3-digit guy. I'm the co-chair. Anyway, as I said, I don't really get deep into the details. I stay broad. In this study, we randomized, it was not a randomized study, but we took about 30 or so HIV-infected people on ART. We brought them in, and we did a beautiful characterization of their baseline, and using certain measuring doses very carefully, we added sirolimus, all right? So an uncontrolled study, single-arm study, baseline evaluation, add the mTOR inhibitor. It was mainly a safety and a PK study. The side effect profile was as expected. You get some mTOR, you get some changes in lipids, glucose, they're manageable. The big problem that we have with mTOR inhibitors in this population is you can get some of these big oral ulcers, which are quite painful, but they resolve when you take away the drug. And I think we saw 1 or 2 of them. But in terms of immunologic stuff, there's not a lot of deep immunology done in people with these drugs, but we did see, as expected, a reduction in the frequency of CD4 positive cells that are proliferating, which is sort of our main target. We wanted to reduce that massive homeostatic proliferation to see if by turning off proliferation, we would get preferential loss of the reservoir size. And we saw reductions in immune activation and proliferation in the CD8 T cells as well. So we saw the T cell stuff that we wanted. Some of the inflammatory biomarkers went down, some went up, but we were mainly interested in once you got through this black box of the immune system, what happened to the reservoir? And this is what happened. So if you look at the frequency of cells that have HIV DNA in it over time, it's pretty stable. And then you go on an mTOR inhibitor, you get a modest effect. And I got into a fight at a meeting recently whether modest was an overselling. But so they said it should be minimal effect. I don't care. Because I've been doing this work for a decade, and no one has ever budged HIV DNA. It's an impossible thing, short of a transplant, to move. And we moved it in the right direction with a nice p-value. So we're happy. We ain't going to cure anyone by reducing the virus, the reservoir this low, but it proves to us, it's proof of concept that by working in these multidisciplinary teams, you can advance the cure agenda and perhaps do something worth following up on. The other drug that we're quite interested in is canakinumab. I mean, I still think the most exciting study done in medicine in the past couple of years for aging in general is the CANTO study. It's a cardiovascular study, so I don't think it's gotten a lot of attention outside of that cardiology world, but this is a study that looked at canakinumab, which blocks IL-1 beta. I think it was 18,000 patients. I'm sure Russ Tracy was involved in it. I know his friend Paul Richter run it. It was a beautifully done study and asked the question, by blocking IL-1 beta, this sort of source of chronic inflammation, can you reduce heart disease? And the impact of blocking IL-1 beta on reducing heart disease was modest. It was real. And it provides the first definitive proof that I'm aware of in vivo that inflammation causes heart disease. Russ, is that a reasonable conclusion? Right. So Russ, you should ask Russ about his stories in 1978 in this little small conference room where they discovered in their heads that inflammation in CRP causes heart disease. And now we have final proof with this beautiful study, CANTOS. But what was nice in this study, which was a shock, is it reduced everything, particularly cancer. Incidence cancer went way down. And all sorts of other comorbidities got better, including all the rheumatologic stuff. And because there's a lot of ID docs in here, you guys are going to freak out. So there was more infectious disease complications, but they were basically manageable. There were a couple of deaths in older diabetics, but the overall mortality rates in the treated arm was lower than the control arm. And I thought this was going to have a huge impact on the aging epidemic. It was going to have a huge impact on cancer, anything related to inflammation, but they ran into lots of problems, I think, getting this through the regulatory process because the primary endpoint, and the only one the FDA cared about, was the cardiovascular stuff, and I think the effect was too modest. But there's a lot of interest in pursuing this, but I think one problem is that the drug is now about to go off patent, and we'll see what happens. But this... And we're working with canakinumab, and Priscilla Shue's got this study where she gave it in a small number of individuals, and we showed that with canakinumab, you can reduce some of the chronic tissue inflammation that you see, and we're going to see if it affects the reservoir. But again, just another example of multidisciplinary work, team efforts, cross disciplines, cross institutes, where we think we can make it some progress here. So in conclusion, older adults, however you want to define it, have unique needs that might be addressed with a cure, but less likely to tolerate. I personally think very powerfully and very strongly that we have to enroll people over the age of 65, but everywhere I go, I get blocked. And I get blocked, and mainly because there are concerns that the older people are going to have more side effects. But I don't think we know that, and we certainly should try to figure this out because this is where the epidemic is going. The impact of age on the reservoir is one of the great unknowns. It's not been well studied. It should be. I hope it gets done. And I think this HIV&Aging has attracted a lot of people who are interested in inflammation and people from outside of HIV. So this is a unique meeting and a unique group of people that I think if we could put our heads together, we can leverage other advances in other fields to make progress in trying to cure HIV. Thank you.